organization at various scales, right? So he's particularly interested in pattern formation, spatiotemporal organization of cells within tissues. His talk today, it's a two-part lecture, will be on collective cell migration. This is the first time I think we will hear about collective cell migration. So yeah, Edward.
So thank you very much. Yeah, thank you very much for the invitation. So I think probably this lecture will be a little bit more about sort of at the blackboard on sort of on background. I'll try to still have a little bit of sort of unpublished research that we've been doing over recent years. And then maybe the second lecture will be sort of reversed with a little bit sort of less background and more sort of research. Let's see where we get to. And obviously, as in everything, sort of feel free to ask any questions you want and interrupt at any point. So I'll start by showing sort of 10-ish slides, essentially, which are pretty pictures and pretty movies to motivate you on why collective cell migration is interesting and cool. And then I'll just review a little bit of some different work at different points of the 20th century on the theoretical effort to try to understand this. So when thinking about sort of collective migration, it's one topic that has been sort of particularly looked at in the context of E. coli and bacteria in particular. And these are beautiful experiments that come back from the 60s, where essentially they're really simple. The idea is that you plate a bunch of bacteria in sort of in the center only, and then everything is uniform. So they're on a plate with some food on top. And what you see is that you could think that things are diffusive, right? You could think that you just have some diffusion of bacteria that sort of go around, but there's surprising structure to it, right? So you see these beautiful concentric rings that are essentially density, they're density bands of bacteria. So it means that sort of there's suddenly sort of collective motion of many bacteria that sort of expand as spatial rings, and there's even some degree of periodicity to it, right? So this is like an incredibly simple experiment, and there you can even do this kind of seed sort of two populations of cells, and you can see that the waves, for instance, sort of kill each other when they meet. So I mean, that's pretty beautiful, right? And that tells you that even in these super minimal systems, there's already sort of relatively complex collective motion and organization. So that's a paper from the 60s. If you play with things a bit more than if you play with things such as the strain or the type of food, it was realized like 30 years later, for instance, in this paper by Madryn and Berg, that you can give rise to essentially any type of pattern. So you can sort of get not only sort of the same rings as they had in the 60s paper, but also sort of incredibly beautiful sort of expansion coupled with, so you still have this sort of global expansion, but then it's mirrored with sort of something that looks like pattern formation of phase separation, right? And you can get sort of all sorts of patterns with different sort of geometries as a function of the experimental conditions, bacterial strain, et cetera, et cetera. So this is pretty cool, right? And this is all to do with, let's say, sort of expansion of a population and pattern formation. So you start in the center, you expand. And so first of all, you have to understand the mechanism of why do you expand as a ring at constant velocity. And you also have to explain why there's all this sort of pattern formation forming. You can also, another model system that people have looked at in the past, which is beautiful, is Dictyostelium, which is a social amoeba that does exactly the reverse. So here again, right, you sit in the center and everything is sort of expands outwards. You can do the reverse experiment where, so this is a movie where essentially this Dictyostelium, these are essentially social amoebas. They can live as single cells. So here you don't really see them, but there's like thousands of cells on that field. I'll play the movie. And under specific conditions, these cells can actually aggregate and form like an organism. So it's a super weird thing which can both exist as single cells, but also exist sort of as but reconstruct an organism. And so this has sort of attracted a lot of attention over the last decade because the movies are amazing. So you start here with basically, so you start seeing, maybe you see a cell like, I mean, the cell is a little dot here. You put some conditions which the bacteria, which the Dictyostelium doesn't like, which force them to aggregate. For a while, nothing is going to happen. And suddenly, it's going to be sort of a pretty big contraction pole. You're going to make this kind of crazy branching tree. You're going to sort of accumulate into one big thing. And at the end of the movie, you form sort of these slugs, which are basically organisms. And these organisms are going to sort of move around and sort of explore space spatially. Which is, again, pretty crazy, right? So this is, in a way, a bit the other extreme of this, where you can not only sort of expand collectively from a single point, but you can also do the reverse, right, where you sort of recondensate to a single point, and that's pretty. I mean, that's pretty large scale, right? I mean, you saw there was thousands of cells. It requires sort of really, it's clearly a collective effect, right? There has to be a lot of sort of collective organizations here. Okay, so the question is, I mean, the question is what drives this? And it's known for a long time, it's been known for a very long time in all of these experiments that active cell migration and directed migration plays a key role in this. Essentially, that cells have a polarity, they exert forces in that direction, so you can think of them as sort of some active particle moving in one direction. And the question is, why do they all migrate towards one direction, right? Why do they, what coordinates their polarity? That's basically the question. This is a similar question that people have asked for mammalian cells. So this is MDCK. So these are MDCK monolayers, and these are classical experiments that were, I think, first done by the Hannezo group, where essentially you model wound healing. So essentially, what you have at the beginning is you have a bunch of cells which are confluent. They had an obstacle, you lift it, and so you're on a 2D monolayer. You're on a 2D dish, basically, and what you've done is you've made an artificial wound, but unlike a wound, there's no inflammation signal, you didn't kill any cells, right? So in principle, there's very little cues that tell the cells that they should sort of migrate towards that wound, right? But still, what you can see is that they're very efficient at migrating. And this is, let's say, the infinite limit to what Mike showed sort of, you know, I mean, Mike was showing sort of relatively small sort of patch here, but you can make it, and you can make it into essentially an infinite half plane, and sort of they're very good at covering the wounds. And so again, there's been a long debate in the literature whether this was, for instance, sort of compressive forces through divisions that would sort of push the cells outwards. I think it's quite clear that in a number of settings, it's active migration, and you can actually visualize it. So this is a paper by some Japanese collaborator, the lab of Tsuyoshi Hirashima in particular. And what they can do is they can actually visualize the polarity of the cell. So this is, it doesn't matter what's green and what's red. The key thing is that the cells exert direction in a polar way, and essentially you can get an insight into the polarity of the cell by making a vector from the green center to the red center. And what you can see here, and this was something that sort of that was alluded to that in plastics, that also that was initially shown by Shavit Repa, is that it's not just these cells here that are in the right direction of migration. It's not just, I mean, these cells, it's pretty obvious that they should know towards that they should go towards this part, right? Because I mean, they have a gap in front of them. But what's quite cool is that even if you look at cells very deep in the bulk, they know they're also pointing outwards, right? And that's again poses a question of collective ordering, right? Basically, what you see here is that it's essentially you have long-range polar order, which in 2D is a non-trivial problem. Yeah. I think it's convenience because you can stain it in live. This is a live movie, so you can sort of have live reporters for it. I mean, I guess you could have centrism. I mean, it depends on the cell type. Some cells have it in the back, some cells have it in the front. The point is that it's a little bit cell-dependent. The key thing is that there's a lot of things you can stain. I think it's just it's for practicality. This is a good way to stain in live something that gives you a vector, basically. Yeah. So this is a big. I mean, this is a big debate. I mean, there seems to be some cells that are more driven by the edge. Some cells that are more that also propagate in the bulk. To my knowledge, I don't think there's a very clear answer. There's not a very clear answer to this. I mean, I think this is also something that Ulrich alluded to. I mean, yeah, but yeah, exactly. They want to, but the fact of whether you have long range order. This is not so. What you can do is that this is millimeter, and you can make it much longer, and it seems to go on and on forever without a clear bound, at least for this cell type. But yeah, so these cells at the edge, they always like so the cells at the edge in basically every cell type, they tend to polarize, right? Because they see empty space and they don't like it, and so they migrate. So the cells at the front. Always, I mean, that's a single cell response, basically. It's not really a collective response. So these always do. The question is, how do you collectively propagate information in the back, basically? Yeah.
So, like, there's two kind of forms of polar order, right? There's like the direction of the cell specifically and where it's moving. Those aren't necessarily given, or are they the same?
So here I'm going to define polarity as the direction that cells move, essentially. So I'm going to define polarity as basically where the direction to which the lamellipodia is forced. So basically, in this talk, I'm going to equate sort of polarity to the direction at which the direction on which you exert your active forces on the substrate. Yeah, exactly. And you can see, I mean, you can do traction force microscopy on this, and you can see that there's an agreement between the traction forces and this measure of polarity. Yeah. No, there are waves in this. I mean, there are waves in this system. That's going to be the part 2 of the talk. It's probably going to be sort of it's probably going to be tomorrow. There are waves indeed. There's also local. So you also see cell divisions. So early on, sort of I think in the first papers of the Silberhausen group, they sort of inhibited cell division, and they showed that essentially most things are conserved. I mean, in this cell type, most things are conserved if you inhibit cell division. So the idea is that this kind of behavior, I mean, after a while, you're going to exhaust yourself if you don't divide because you're going to run out of material. But to some degree, this response is not division dependent. Yeah.
I assume that this is like a time frame of 10 minutes or so.
I guess.
The cell division is mostly very fast, and you would never catch them right in the cell division.
Well, you sort of see, you see them a little bit like here, for instance, there's some cell dividing. I mean, yeah. But yeah.
The round up become very large.
Yeah. These are the flashes, yes. I think so. Yeah, sorry. Sorry? Yeah, exactly. I think it's. Yeah, I'm pretty sure it's cell division. Yes. Yeah. All right. No more questions on this. All right. So then, the question here essentially is that you have long range order in the system, and the question, I mean, and so one of the key questions I want to sort of ask in the talk is how do you get that, right? And so there's essentially two things. I mean, this very question has been, I think, tackled by two very different communities over many years. And I want to sort of go back and forth a little bit between the two. One is, which would be a bit more the topic of this lecture, is more about understanding what aligns cell polarity, and it's more about thinking about reaction diffusion equations. That's more prominent in the mathematical biology literature. And the other one is sort of statistical physics and the ideas around flocking, which I think Christina mentioned already, which I'll talk about a little bit sort of in the second part. On the first part, there's a very, very old sort of, I mean, there's sort of over a century old knowledge that one of the key things that aligns cell polarity is gradients of diffusible molecules. So there's a lot of diffusible molecules. I mean, there's a lot of things that the cells sort of sense, and where the cells can make a directional measurement and sort of see and move in the direction of a gradient. The classical thing is chemical concentration. Over recent years, actually, there's been a huge generalization of this. We are realizing that it's not just diffusible molecules, but also sort of substrate stiffness, electric fields, friction. There's a lot of, my point is that there's a lot of signals that if they're graded, can align cellular polarity. That's going to be one of the key ingredients I want to add here. But of course, the classical view of the field is that there's some pre-patterned gradients. There's essentially some pre-patterned gradients which the cells follow. But in all of the examples that I've shown you, this can't be right? Because if you think about this social amoeba, no one made pre-made a gradient, right? If I showed you these MDCK monolayers, nothing pre-made a gradient. So sure, you can say, okay, there are some gradients of molecules that align cell polarity, but that in a way just displaces the question to ask where does the gradient come from in most situations. And so one of the key things I want to talk about today is the idea of that comes again from mathematical biology and reaction diffusion, the idea that there's feedback between cells and their own diffusible fields that they can follow. And basically, this one of the most spectacular example of this, I think, is work from Rob Install over recent years. There's essentially a really interesting idea that has popped up. I mean, that is again very old, but has popped up over the recent years, is the idea that cells can actually eat up their own chemoattractants. And why is it cool if you eat up your own chemoattractant? Well, that's a mechanism for collective movement, right? If you imagine that you have like everyone is on this side of the class and your chemoattractant is uniform, if you also eat what directs you, well, in the zone where there's a lot of people, there's not gonna, you're going to deplete your chemoattractant, and then suddenly you're going to migrate towards open space, right? So that's a very, very generic mechanism of the idea of self-generated gradients that you can use to migrate. And so there's this beautiful paper by Robin Stoll where they took sort of both dictyostelium and cancer cells and they put them in mazes with uniform gradients. So it's basically a maze, which is sort of microfluidics, where here these are all the channels of the cells, and you see that there's dead ends. It's basically a labyrinth, and they need to find this end here. And everything is flooded by a chemoattractant, so everything is uniform, but you put cells only here on this side. And if you look at the movie, what happens? So again, there's no pre-patterned cues, but what's amazing is that if you put cells in there, see, cells can essentially collectively solve mazes. And that's a collective effect. If you put very few cells, it doesn't happen. And so you see that actually, if you look at the first, and this is really not a trial and error thing, right? If you think about yourself in a labyrinth, you're going to sort of try things at random. You're going to bump against a wall. You're going to go back, right? That's going to be much more of a random walk. If you think about these cells, if you look at here, very few are going to go down in this trap, right? Almost all of them are going to go early on towards the side. So you see what I mean? There has to be some long range queue. Yeah. So that's my point. No. So initially there's no gradients. Everything is uniform. And so one of the idea, and we'll go through a little bit sort of the type of derivation. The key idea from this paper is essentially that the cells reshape their gradients. And so even though the gradient of diffusible molecule is uniform initially, because you have essentially, because cells, because cells here start eating up the gradient, then through diffusion, your things, you're going to sort of, you're going to reshape a gradient. And the point is that if you have a reservoir of things here, you're going to get you're going to get sort of a stronger gradient from here to here than here to here. Essentially, it's a diffusible. What you eat, essentially what you eat in front of you diffuses fast and sort of creates sort of hints at the path towards the open end, basically. We'll go. I'll go a bit more in the derivations later on. Yeah.
Like reduce density, we say one at a time.
Yeah. Yes, exactly. And we've also worked on it. I also mentioned we've done that. So essentially the degree of polar order or the degree of sort of how good you are, and is enhanced by having more cells. It's a collective effect. Yeah. Why did they go up? So because everything is uniform, and basically the idea is that here there's a big source. Here it's a reservoir, so it's a big source of attractants. So here you start eating up things, and so things diffuse. So you start eating up things, and things diffuse. But here there's no source there's no source of molecule. Whereas here there's a source of molecule. So basically, you start essentially you start making sort of a flat profile here, and you can build a gradient from here. And so that's the core idea. Again, I'll try to make this a bit more precise later on, but that's the idea of that paper if you're interested. Yeah, yeah. So that's a big question. That's less clear. They do it up. What you can see is that the cells do it up, and the cells do it up. And it's a finite resource. And you might have noticed that the cells going up afterwards are less precise than the cells going on afterwards. So there's also some exhaustion in the system, which is sort of, so there's some time-dependent aspect to it, which complexifies the analysis. Yeah, yeah. It's much slower. These are cancer cells, so I would think sort of tens of microns an hour, I think usually you can sort of treat the gradient formation as sort of fast compared to the movement of the cells. That's the general idea. So that's why the sort of the diffusion. The diffusion of the attractant that they eat is sort of a fast signal compared to their motion. I think that's the general idea, but I don't know exactly. Yeah.
Are there other interactions between cells, or is it only via the gradient?
So that's a good question. It doesn't seem like there's a lot of interactions. You see that they're not touching that much, right? It's very hard to imagine at these densities how they could sort of make sort of, you know, how they could flock or do some kind of long range order. So I think it's no, there's not much. I mean, what you can also visualize is that if you don't have any diffusible attractant, nothing happens, which is a good. I mean, you don't need to invoke this to explain the data. Let's say, of course, you could imagine other more complicated things, but you know, it's not. Yeah. 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 Yes. So I don't know. So I don't know exactly the detail. The point is that it's a 2D. It's a 2D sort of. It's essentially a quasi 2D macrofluidic channel where the molecules can only come from this reservoir or this reservoir. So nothing can comes in and out of plane basically. So you can think about it as sort of. You can think about it as sort of a gradient. I mean, diffusible. Essentially, what you solve is you solve a diffusible equation with some sinks with the cells behaving as sinks. No, everything is uniform initially. The cells make their own gradients. Everything is uniform initially. So these are cancer cells and dictostelium. Either. I mean, you know, I would refer you to this paper. I don't know. I don't know to this level of detail. What's the cell line? Yeah. All right. All right. So that's the one idea that I mentioned is about sort of the one idea I'm going to mention here is the question of diffusible molecules. And actually, maybe I'll stop here because I'm already sort of 20 minutes in, and I mean I wanted to talk about flocking, but actually this is going to be more in the second part. So maybe I'll go to the board now. All right. So the question is now is what are the minimal things that you want to what are the minimal description that you can make out of these cells basically, and let's go again to some really minimal thing. Let's say that you imagine you have something in 2D, so you start with a super high concentration of attractants here, and let's say that these sort of radially move outwards. You know, let's go back to the first experiment where you had, you know, your radial expansion of bacteria. Well, the simplest thing is that essentially there's a lot of bacteria associated to this, so you don't really care about individual sort of bacteria. What you care about is a concentration field, right? So you pass everything to the continuum limit, and that's the concentration of cells. And let's write it as a function of, because I'll do everything. I mean, you can do it in multiple dimensions, but let's say that it's spherical, and so let's write it as a function of one direction. So what you want to write, essentially, the simplest thing you can write out of this is essentially writing reaction diffusion equations. And reaction diffusion equation is nothing more than a conservation equation on your the concentration of bacteria. And so the broadest thing you can write for this, is essentially something like this, where essentially a conservation equation can always be written, right, as if you look at what changes the local concentration of your bacteria, it changes due to a flux J, which is essentially sort of so it's a flux, and there's a non-conservative, so that's a conservative part, and then you have a non-conservative part, which is essentially because these are cells, they can also divide, right? So there's two terms here, growth essentially and advection, and the question is what are the minimal ingredients that you need to give rise to this kind of sort of collective motions of cells? And so this has a very long history. I'll actually sort of even though I just told you that everything is really important on cell motility, let's forget everything for a while, because actually I think it's a very instructive example to go back to what can happen without cell motility. So the simplest thing that people have sort of done for this, which has a pretty illustrious history, is something that was independently discovered by Fischer and Kolmogorov and collaborators in 1937. So it's the same Kolmogorov as the one that you know from turbulence, did a lot of things. And this was really thought as thinking about population dynamics. And the idea of this is essentially saying, let's forget all about sort of active motion. So if I forget all about active motion, I hope I don't need to convince you that the simplest thing that shows up is just diffusion, right? So fixed law. So I just said that essentially each of my cell does a random walk, right? So at the continuum level, that's just a diffusive flux, fixed law. And then the other assumption from Fisher is saying that my bacteria can reproduce. So if my cells can reproduce or my populations can reproduce, the first term is saying that they divide so exponential growth. So if they divide at a given time rate tau, you would have FC equals C divided by tau. That's just exponential growth. That's not going to behave very well because that's clearly going to explode, right? That's exponential. And so what's the simplest term you can stabilize? What you can say that, well, no cell can grow to infinite density. There's always going to be a saturation term. And a simple saturation term you can write is something like this. It's essentially logistic growth. It's saying that basically cells divide. Sorry, can I move this? No, I cannot move this up. Right. Okay, that's it. So that's the simplest thing you can write, basically saying that you have a carrying capacity that cells at low density grow exponentially, and then at some point they reach carrying capacity at C0 and they stop growing there. So that's really the absolute simplest thing you can write. If you think about these things, and it's not completely trivial. I mean, it's a classical result, but if you dezoom from, I mean, if you've seen it before, great for you. But if you think about it, it's not so trivial what's going to happen, right? Because if you think about this term only, if I say I have a delta function, so if I say that my initial condition is just a delta function, right? Something like this, or I mean, let's normalize it to 1 because it's things. So if you take this term here, things are going to grow as square root of t, right? It's just going to be diffusion. So the way that the whole thing spreads is going to be as square root of t. This is a bit less clear, but if you start from something which has fat tail, this tends to be exponential, right? I mean, because this is exponential growth. So so one tends to become sort of sublinear, and one is superlinear. So what happens when you have both? Well, I'll just run you through quickly through the derivation because I think it's actually useful and it's something that shows up. Yeah, sorry. This is tau. So this is a time scale. This is a time scale. So if you want to make things dimensionally correct, this is concentration over time. So if you want things to grow exponentially, you need a time here, and that's basically the time of the division time of cells. Yeah. Okay. So what do you do? So then what your Fisher essentially what your Fisher equation, right, is sort of C tau 1 minus C. And let's forget about this and let's say that let's non-dimensionalize my concentration, my C0 to 1, because I can do that without loss of generality. What happens? Well, what you can do numerically is you can see that this shows a very similar behavior to here, which is as essentially if you start as x from some delta function, essentially what happens at a concentration of 1, essentially you see that there's a traveling wave that forms at constant velocity. That's a numerical result where this is a super simple model that gives rise to a traveling wave or a traveling front at constant velocity. The way you can show that very briefly is if you think that you see a traveling that you have a traveling wave, what it means is that I should be able to go in a co-moving frame, which means that, right, I can redefine my coordinates. I can redefine my coordinates as y, right? I can have a collective coordinate y, which is just x minus ct, c being the speed of propagation of my wave, right? And if I have a traveling wave, I can express everything as a function of just y, right? Which means that if I re-express this equation, I'm going to get, sorry, I'm not going to write it c, it's very confusing. V is the speed of propagation, which means that essentially you're going to get something which looks like c dot dot plus c tau 1 minus c. Right, so this is because essentially my second derivative with respect to x is just exactly my second derivative with respect to y, and my derivative my first derivative with respect to time gets a minus velocity here showing up in front of that. Okay, what do you do with that? Why do you have a velocity that shows up? Again, it's not the key part of the course, but I think it's kind of nice. One simple thing you can say is that you can also, like, essentially, if you have a front expansion, right? You can look at the edge of the front, and at the edge of the front, my concentration is very small, right? So I can say that my concentration is very small here, which means that which is very practical because it means I can linearize my equation, right? I can mean that I have minus v c dot equals d c dot c over tau, right? I can forget my non-linear term because I'm in the front and my concentration is very small. This is nice because this is just a linear ODE, right? So what it means is that this profile at the front, that's something very generic in this kind of reaction diffusion equations. This is going to decay exponentially, right? And I can just get my characteristic equation by plugging this into that, and I can get sort of d squared plus v lambda plus 1 over tau equals 0. That's my characteristic equation where I can get the length scale of decay of my front. Why is this useful? Why is this useful? Not just for that. Well, you can actually try to solve this equation, right? And you can try to get lambda. And essentially, what lambda gives you, right, is that this is minus v plus or minus square root of v squared minus 4d over tau. And this is actually quite cool. I mean, and again, this is something that shows up very often in this kind of reaction diffusion problem. What you can see is that not every velocity, not every sort of lambda is authorized by this. Right? Sorry, not every v is authorized by this, because you can see that clearly, if I want a front to propagate, I need this to be I need this to be real. And clearly, if I have v, which is less than 2 square root of d over tau, this term is going to be negative, and I'm going to have some imaginary part in there, and that's not going to be nice, right? So what this means is that this means that actually this is a criteria for wave selection, which tells you basically that no wave of velocity less than this criteria can actually propagate. So this tells you already that from just linear analysis that only specific velocity speeds can be selected. And it turns out that if you sort of look at the back of the wave, you can actually show, and that's the key result of Fisher KPP, that this is an equality essentially. That if you have some not too weird initial condition, essentially you get linear wave propagation with this kind of thing. And it's kind of, again, to go back, it's actually kind of a nice result because you see that the linearity of this really comes from a compromise between your diffusion, which tends to make you as a square root of t, and your sort of tau that tends to grow exponential, right? So it's a little bit of a compromise between the two. Yeah. Sorry? Well, I mean, it's not physical. I mean, it's not really physical if you get it. I mean, it's not something you would see in linear. I mean, basically the point is it's not a wave. I mean, it's not something that would show up in simulations, right? So what you see in the simulations, if you do a simulations of this, it's this velocity that shows up, and it's this one that's selected essentially. There's only a certain sort of range of speeds that are authorized, and it turns out that if you go at nonlinear order, there's a strict selection criteria. Okay. So I've told you all of this, but okay, you might say sort of, but I don't care about. But here you didn't put any sort of active migration, and you're right. And actually, but I think it's kind of nice to compare and contrast this model to the one that now invokes directional migration. So now let's go to another type of model, which is essentially which is something that is, I think, sort of a little bit too unknown. Sort of everyone, sort of, for instance, knows about Turing patterns or something. This kind of Keller-Zegel instability is a bit less known, even though it's absolutely beautiful. And it's something that has a long history. So there's some prototype of this by Pat Lack in the 60s, but the key model of this really comes from the 70s by mathematicians of Keller and Zegel. And this also forms the basis for subsequent things, for instance, in the 80s by George Oster and Murray, for instance, if you know about it, but it's not so important. So what is the Keller-Zegel model? The Keller-Zegel model is essentially the simplest variation of this that takes into account active motility. So what do you do in the Keller-Zegel model? You take exactly the same structure of equation, but now I want to put active motility. It's going to be conservative, right? So I have to put it in my flux. And the simplest way to write it is to keep my diffusion, my Fick's law, but add advection into this, right? That's the simplest thing. That's very classical in any conservation equation, right? In your flux can have a diffusive part as well as an advective part. And so to stay simple, actually, in principle, of course, you could combine this new term here with also the fact that cells grow and divide. But actually, the original model of Keller-Zegel, and I think it's the simplest for clarity, says let's forget all about division. So let's put my my growth term to 0, and let's look at what advection does exclusively. That's much simpler. And there's a huge body of mathematics and PDEs that has looked at combining the two in every possible sort of way. So if you want, there's like probably thousands of papers on this, but we're not going to go into this, and we'll look at the limiting cases. So the key thing now in this model, once you've said that, the problem is that you haven't said much because you've introduced a new variable, which is v. And now the key thing is what you couple your velocity to, right? So if you think about cell velocity, you need to write an equation for that. And so again, the key intuition from Keller and Zegel is to say, well, my cells go in the direction of my diffusible field. So in addition to C, I'm also now going to have a field X of T, which is my chemoattractant, which is sort of these diffusible molecules that attract cells. Chemoattractant. And so the key idea is that we're going to, for now, forget all about the physics, like I hope to go back at the very end, but we're all going to forget about the physics of cell polarization, actin, everything that says polarity. And the simplest thing is to make a choice, like, for instance, velocity being sort of equal either to gradient of n, or another choice in the literature classical is essentially sort of relative gradients. That's basically the idea that sort of cells migrate in the direction, either in the direction of absolute gradients of your diffusible molecules or of relative gradients. And there's a lot of literature of chemotaxis that has looked at the single cell level, what are the mechanisms, and you can make arguments for either. It depends exactly what's your sensing mechanism. I'm not going to enter into the detail because I want to stay at the continuum level and sort of write it as PDs. So that's one thing, right? So essentially, if you do that, you have your equation for a C that looks like so you get one equation for your concentration now, which has this diffusion field and which has this advection that has sort of gradients of molecules in here. And very clearly, I need to write some equation for n as well. And so the Keller-Zegel model is to write a very generic equation for n, where basically n sort of diffuses and then has an arbitrary function g, which is C of Cn, which is basically CNN. And then the question is, okay, that's a bit sort of everything you can ask. And then the question is, okay, what do I write? For this, for instance? All right. So the original model of Keller and Zegel is to say that okay, if I have, for instance, if I have, let's say, a front on this side, if I have a front of cells that wants to migrate in this direction, the reason it could move is if the cells eat the thing n, and so you want your n to be like this, right? If your cells eat your diffusible molecule, it's going to look like this. It's going to be quite cool, right? Because you're going to sort of move forward, then eat, then reshape your gradient, move forward, eat, reshape your gradient. And so the simplest thing you can have for this is writing something like minus Cn, right? Saying that basically your n is degraded proportionately to the number of cells here. That's like the simplest order equation, where you say that basically you're at the continuum level, where you say that essentially your cells eat up your chemokine, right? Okay, is that clear for everyone? Okay, so what happens in this context? So now you have a closed set of equation. You can do exactly the same trick, where you can sort of go in a co-moving frame and rewrite those equations here. And what you get, you get that sort of minus v c prime equal d c dot dot minus c. Let's make that constant, c gradient of c n prime n dot, and then you write sort of also v n dot equals d n dot minus c n dot. Again, I'm not going to go into the detail of this, of deriving exactly which velocity equation you write. If anything, actually, the fact that there's no the fact that this equation is conservative actually helps you. So there's no growth term. Why does it help you? Is because you can integrate this once, right? If you don't have if you had this f of c, you can't integrate once this equation, but here you can. And so this actually simplifies quite drastically this whole thing, right? Because you can essentially get rid of what indices because of the boundary condition that at x infinity you don't have you have c equals 0. Absolutely no reason. Thank you very much. Exactly. Yeah. Oh, I'm sorry. Yes. I'll try to write a bit bigger. Yeah. So I don't want to. I actually don't want to enter into detail of this. I'll give you the answer. I mean, it's if you want, you can actually go in the detail of these things. I mean, you can solve it. The simplest way to solve it analytically. So in Keller and Zegel, essentially, the simplest way to solve it analytically is to say that this is essentially zero. That this is essentially 0, and then you can reinject this into this. The key idea that shows up, and it's you can make a number of choice for the So that's the simplest thing where if you just have if the n degrades on its own, you would have a minus n term, right? That's radioactive decay. What I'm saying is that it's not your molecules are not spontaneously degrading; they're degrading because they're eating. So if you think about it in terms of chemical reactions, it's like one cell meets one diffusible molecule, and you remove the diffusible molecule. So it's like essentially sort of the first order chemical reaction between sort of one cell and one particle. I mean, again, there's a huge mathematical biology literature. Like if you can put an exponent q here, and you can look at how q changes your whole result and everything. But I'm not going to go into this. The key result I want to get from this is that this also admits traveling wave solutions, and the qualitative thing that the key thing is that essentially you get traveling wave solution is essentially you always end up with some version of that condition where essentially what you need chi, which is the efficiency of going up the gradient. So chi is how efficient you are at migrating up the gradient. So you can see it as sort of this is essentially sort of your advective term. This is how efficient our cells are migrating, and this is essentially your diffusive term, right? And so again, qualitatively, this makes a lot of sense, right? It's because this is basically, if you want a traveling wave, the point of the traveling wave is that it's something that has no dissipation, and so it sort of makes sense, right? That if you had only diffusion, it wouldn't work, and so you need essentially your advection term to be larger than the diffusion term, and that's again something very, very generic that shows up out of this. And so what it ends up, right, is that if you integrate these equations again, what you're going to start, if you start from cx of t, cx of 0 and being like this, and n being like this, or let's just do this, what you will have, in time is that n is going to be eaten by c, so c is going to start migrating upwards, but then it's going to eat even more n, so n is going to move, so n is going to be degraded more and more, so it's going to move towards the left. And as n moves towards the left, c moves towards the right, and that's the mechanism of this traveling these traveling waves. Okay. There's a really cool thing with this is that you can actually so this can explain relatively well why you have a front expansion, right? In this first experiment I've shown you, where so if you start from essentially a delta function of bacteria, they migrate upwards. You can ask, what about my social amoeba, right? Which was doing the reverse. My social amoeba was condensing into a single point. What was happening here? Well, the cool thing is that you can do exactly this, but you can just change now your g. Sorry, I'll write a bit bigger as g. You can say that g of cnn is instead c minus n. Why do I want to do that? Well, if you think about aggregation. Clearly, aggregation cannot happen if you eat your own chemoattractant. But if now cells, instead of eating the own attractant, produce it, then that's a mechanism for attraction, right? Because now regions of high cell densities produce a lot of chemoattractant, which makes even more cells migrate towards them, right? So you end up having something where you can essentially rewrite the exact same model, but instead now C, instead of being negatively impacting the concentration M, is now positively producing it, right? And if you do that, so maybe I'll go. Maybe I still a bit of space here. I'll go here. Actually, it's a bit better. So what you can do now is, so you have your equation on C. You have your equation on C, right? You have your equation on N. And I'm writing plus C over N. I mean, as you see, this is based on non-dimensionalization, right? I have some units, and I'm not going to care about them. Yeah. Well, so if I do C times N, it's not going to work because my N is going to blow up, basically. You still need some, I mean, you know, if I have C times N, I'm going to have blowing up solutions, right? Because you need, I mean, if your cells produce N, you need some sort of, you know, radioactive decay. You need some spontaneous decay of N, otherwise things are going to blow up, right? So that's why you need some negative, I mean, you know, you need some negative term, basically. So that's why, I mean, that's the simplest thing you can write to linear order that has the right sort of, that doesn't blow up, basically. Okay, so I think it's kind of useful to, if you analyze this, to just do a very simple linear stability analysis to see what shows up in this. So the idea behind this, right, is, I'm sure you're familiar, is that you say that C is some small perturbations and N is some small perturbation, right? And I want to look at sort of, and I want to look at how these grow as a function of time, and so if you do that, I'll actually give the solution because I think it's a little bit more instructive. It's actually easiest to neglect this. Let's say that N is a fast variable. What you get this if you pass this through Fourier if you look at this in Fourier space, is essentially that delta N is equal to delta C dnq squared, right? So basically, what this tells you is that N follows essentially C up to some sort of operator that sort of makes it diffuse. And what you can do is you can reinject this to linear order in this equation. And so what you're gonna end up with as an equation for dt of C is you're gonna get some diffusible. You're gonna get some diffusion here. Sorry, I'll write it directly as in Fourier space again. So you have some diffusion onto minus here. And then you have a term which is kind of nice, which looks like so you have a term here, right? So here, this is in Fourier space. So this is d of q squared. So right, this is just this is always stabilizing. I mean, diffusion always stabilizes you. If you think now about the modes, if you look in terms of dispersion relation, this is always going to stabilize you, right? But here, if you look at this term, this is a destabilizing term, right? This is a term that's positive, and it means that this entire thing can be positive, which means that specific wavelengths are going to have an instability, and you're going to grow. And so you can actually look at this dispersion relation of this. So you can ask, do my perturbations grow or shrink? And so if you look at high Q, this thing is going to disappear, right? Because this is, if you look at high Q, this is going to be of degree 0, whereas this is of degree minus Q. So it's always, so this is always stable at large Q. There's no problem. But if you look at Q equals 0, this doesn't matter, right? If you look at Q equals 0, so if you look at Q equals 0, the way whether this starts positive or negative is gonna really depend. This depends directly on the sign of chi minus d, right? If my advective term, if my sensitivity is positive, I can actually this whole term can be positive, and it means that I'm going to grow and I'm going to get pattern formation. Whereas if my advection is slower, is less than my diffusion, I'm going to shrink. So again, you see, it's kind of nice. You see that it's exactly, even though I reverted the sign and I'm now looking at pattern formation instead of sort of expansion, I have exactly the same criteria where basically I'm going to get an instability, I'm going to get an aggregation instability if my advective term is larger than my diffusion. And again, I think this is even more intuitive to understand, right? If you think about it, if I start making a layer of foci of bacteria that produce their own attractant, they tend to diffuse, which is a stabilizing term, but then they tend to attract but because they produce a chemoattractant, they tend to recruit more bacteria towards it, which is a destabilizing term. And this is actually quite interesting because what shows up very often in these kind of equations is essentially that advection shows up as an antidiffusive term, right? Essentially, up to this term, which I don't massively care about, my advection plus the fact that I'm producing my own attractor shows up as a negative diffusion, which is of course an aggregate. I mean, something that tends to aggregate things rather than spread things, right? So, yeah. Yeah. That's a good question. I suppose you would actually. I'm not completely sure. I guess you can. Yes, probably. I would think so because actually, in Dexoselium you get waves right of cyclic AMP. I'm sure you. I mean, I'm sure you can get some waves in some regime of parameters. That's a good point. I would think so. Yes. I actually don't know straight away. Yeah. Sorry. So this is extremely general because this is a linear stability analysis. So basically anything that isn't, I mean, anything to linear order, which is sort of positive in C, which is positive in C and negative in N, would be the case, right? I could make something very generic as long as my cells produce the chemoattractant and the chemoattractant is degraded itself. Basically, to linear order, you're going to get exactly the same thing. Then, of course, as in many of these cases, the nonlinear terms can do a lot, select different types of patterns, especially in 2D. But this is something extremely general. Yeah. 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 Yes. Excellent. So that's my next page of notes because I can't. Yes. So even though it's not completely related, I can't sort of resist making the. Yes, absolutely. So I can't, because Ulrich already introduced these things, I can't help but saying that actually this is exactly, I mean, and this is quite beautiful about these equations. These were developed again for sort of multicellular systems, but it's exactly the same instability as you get in isotropic active fluids. And the reason for this in isotropic active fluid is exactly the same reason. Is basically if you think about my actomyosin, so if you think about sort of an actomyosin gel, right? If I have essentially my contractility is going to depend on the concentration of the gel, right? So if I have more actomyosin locally, I have more contractility, which means that I have a mechanism where you advect, where you go from regions of low density to regions of high density. And if you think about this, it's exactly the same mechanism, right? And actually, okay, I'm actually going to make it explicit. If you write one-dimensional active gels, so what you write usually is you write exactly the same equation. So now rho is by density of actomyosin, but it doesn't really matter. So the simplest thing I can write in 1D is again a reaction diffusion on this, right? And now because it's an active gel, I have a slightly more. I actually don't write something arbitrary for V. I'm going to write force balance. And so because I have a friction with the substrate. What force balance is going to read I have some friction, right? So force balance reads like this, and then and then my stress is a mix of my stress is going to be some mix of viscous term here plus some activity. So this is something that sort of Ulrich alluded to. So basically, that's my constitutive equation for an active gel, for a viscous active gel, right? I have a viscous term, and I have a term that's actually the negative of what you usually have, right? If you think about pressure, if you think about the pressure in a classical gel, you would have a minus here, right? But here you have a plus, which is a contractility, which is this active contractility. So if I plug this in here, so the first thing I can do is I can recognize. So this is so this means that this is eta dxx v plus x dx of rho, right? This is just injecting. This is just injecting this into this. But the first thing I can do is I can recognize that in my active gel under friction, I have a linear relationship between V and my gradient of stress. And if I rewrite this equation by replacing V, what I end up with is I end up with dxx of rho equals my dx rho dx of sigma, dx of sigma over zeta, right? And this is exactly the same equation that's on the right, right? Basically, what it tells you is it tells you that stress in your active gel equation plays exactly the role of the of the diffusible field, which makes a lot of sense, right? Because what the role of the diffusible field is to sort of align your active migration towards, and what stress does is to pull you is to pull the gel towards high densities. So basically, it's literally exactly the same thing. And if you write your equation on stress, you're going to get exactly this. I mean, if you rewrite this equation instead, I mean, usually people write it on V because it's more convenient because you want to write an hydrodynamic equation based on displacements, but you can rewrite your equation based on stress, and you get exactly the same thing as Scalar-Zegel, because basically your gel produces stress, right, and consumes it. So it's kind of nice actually, and again, this is exactly the reason for active migration. In active migration through actomizing flows, what you end up with is a spontaneous instability where all of the actin flows towards the back, and that's what propulses cells. So it's a type of sort of it's a type of coarsening when you end up with most of your actomycin towards the back, and with constant flows that propel your cells outwards. And that's exactly the same. That's exactly the same sort of physics as why you get aggregation in dextrostellium, just replacing essentially a stress field by chemoattractant field. So it's something, it's not directly related to what I'm going to talk about, but I just wanted to mention it because I think it's pretty neat. Sort of, it shows sort of pretty neat universal behavior across sort of subcellular and multicellular scales with the basic physics of these vective equations, essentially. Yeah. Yeah.
So you're looking at that the mechanism of motion.
Yeah.
That cell division can induce some sort of a pressure, like growth-induced pressure that can drive migration.
Yes. Yeah. So I mean, you can combine all of. I mean, you can combine all of them, right? And people have done this where you have gradients of pressure and you sort of you're pushed. I mean, you can write, of course, a much more general theory out of that. That's true. Yeah. And people have done this a little. I mean, people have done this a little bit because usually, typically for bacteria. So people like. So this has been revisited, for instance, by people like Thierry Roy and Emonet. And there, for instance, for bacteria, very often you can't really separate the contribution of growth and the contribution of advection. So you need to write both terms and then to evaluate it in a data-driven way. So this has been revisited by quantitative measurements over recent years where people have had to put both components. That's true. Yeah. All right. Any questions? I was gonna maybe go towards. I wanted to talk about flocking, but actually I think I'll do that next lecture, and I'll show you a little bit of research now. A little bit of research. So maybe I can pull down the. Yeah. Sorry. Yeah.
So with the second equation, there's a time derivative factor, which the second term is proportional to the product of the C times N.
Yeah.
And so I thought I'm confused about the sign in front of that term.
Sorry, which one you're?
Second equation in the upper left, the time derivative of N.
Yeah.
Plus C times N.
Oh, sorry, C minus N. Sorry, that's C minus N. Sorry, I should have. Sorry, no, it's yeah. N spontaneously degrades and is produced by C. Sorry about that. Sorry, I should make it. Sorry. Yeah. Right. All right. So I have half an hour left, so I'm going to sort of talk a little bit, or give you some examples where we've applied these kind of ideas to experimental data. These are a series of papers, for instance, where, as I said, the idea of sort of self-generated gradients driving migration, this comes back from Keller-Zegel, so this is a very old idea. Oh, sorry. Yeah. Yeah.
I thought that was in the sense that you could reaction at low.
Okay, so a few things. So blow-ups are dimensionality dependent. So blow-ups, I think to my knowledge, don't happen in 1D usually, and are sort of generic. I mean, are sort of marginally stable in 2D and sort of always unstable in 3D. So usually, what you do is essentially you write, but they get taken care of by nonlinear terms basically. You can always stabilize. So actually, in the original active gel equation, sort of in the original active gel equations of Bois and Julie Scher in PRL 2011, they actually write a nonlinear contractility that saturates. And the fact that contractility saturates, actually in 1D it's not important, but in 2D it prevents scalar-Zegel blowups. Yeah. So that's yeah. I mean, the Keller-Zegel literature is a bit weird because the original terms were like just minus C, not minus C times N, and so it means that the contribution can become negative, and there's a lot of weird, there's a lot of weird things that have been fixed since then. I mean, it's a messy literature. Yeah. So what I want to tell you a little bit about is sort of more concrete applications of this kind of concept. So as I said, the idea of cells migrating by self-generating their gradients is a very old one. It comes back from Keller-Zegel, but it has stayed sort of largely untestable in many systems. And recently, this has been sort of this has gone back into trend over the last few years, in particular because people have been able to manipulate biochemically the different studies. So there's a few papers here, for instance, that have all identified self-generated gradients in different things. So this is like the lateral line of zebrafish. This is the paper I mentioned that's solving mazes in dictyostelium and cancer cells. These are work that we did on zebrafish gastrulation, and this is work that we also did on immune cells. And so again, here, for instance, what we had shown with the group of Andrea Paoli in zebrafish migration is essentially what the mesoderm in zebrafish is initiated at the border. So basically, all of the tissue is in red, and you make the mesoderm at the bottom. And that's a very generic thing. Like as soon as you make one of your germ layers, the mesoderm, you make it in a boundary in most animals, and then it needs to migrate up. So it's well known that these blue cells need to migrate up all the way up the embryo. And it was not very clear what was the key. People had been trying to identify the pre-pattern queue that was giving rise to this for sort of decades. And what we realized is that it's not a pre-pattern queue. Basically, you have like a uniform queue, which is a diffusible molecule that's produced by every cell in red. So initially, you have a uniform field, but then your blue cells, your mesoderm, express a receptor that eats up the red molecule. And so it's this. So what we found essentially is a paradigmatic sort of Keller-Zegel model, where essentially when your cells differentiate into the mesoderm, they acquire the ability to eat up their own chemoattractant, and so they migrate very robustly sort of up the embryo. I'm not going to talk to you about this because this is published. One of the key things I want to mention is that in all of these papers, it's a single cell type interacting with a single attractant. And very often, in many different kind of biological situations, it's not going to be the case, right? You're going to have multiple cell populations that can interact in complex ways, right? In particular, what I'm going to talk about is the immune cell response. And there's not like one immune cell type. There's tons of different immune cell types that need to migrate towards the wound. And so the question that we had is, can we generalize these ideas to multi-component analysis where you have multiple cell types? And this is also related to what people have been doing in active matter. For instance, Ramil Goleshanian, by looking at mixtures of particles. I mean, there's some links to some synthetic active matter that I'm not going to enter too much into the details. And the question that we had is essentially, can different cell types communicate their directionality to each other by interacting only through a diffusible signal? So where the cells don't really interact with each other, but only interact through their interactions with a diffusible chemokine. And so this is actually a theory work that sort of is ongoing. Actually, we want to put it on the archive in the next few weeks. So it's great to have your feedback. So it's all done by an outstanding postdoc that's now starting his group in Sheffield. And all the experiments were done by the group of Michael Six, in particular, is part of the PhD of Zana. And so what's the question? The question is that we had shown that in a previous paper, that one specific immune cell type called dendritic cells were exactly a Keller-Zegel type, which means that they could eat their chemokine, so that they could essentially shape their gradients and respond and migrate in the direction of the gradients that they had shaped. But what we had in some supplementary information is that it seems that many other immune cell types cannot do that. So if you put dendritic cells in a uniform field, they can shape it and migrate directionally. But a lot of immune cell types are not able to do it. And so then the question that we had is, it seems a bit, I mean, it's a bit weird, like why is it like only a specific cell type that can reshape its gradient? But then we thought, well, anyways, immune cell migration sort of relies on many different things. And so maybe it's just metabolically efficient to have a single cell type shaping the gradient, and maybe all of the others could surf on the gradient of the others. The idea that the others could sense but not really eat. And that's a bit of a, I mean, because anyways, if there's one cell type that makes the gradient, maybe you don't need every other cell type to do it, and all the others are sort of free riders that can sort of piggyback on that gradient. And so we wanted to ask, okay, first of all, does this happen? Can we explain this theoretically? And are there optimal ways to couple different cell populations through self-generated migrations? So the kind of experiments that inspired us is, the key of the model will be to have two cell types. One cell that can both eat and sense their chemoattractant, and one that cannot eat but only sense, right? And this is based on these kind of experiments where essentially they make a big blob of sort of both of two types of immune cells, T cells and dendritic cells, and they have sort of a uniform field of chemoattractant, and then you can see what happens. And what happens essentially, you have quite a nice expansion where things sort of migrate outwards. And I'm not going to show you all of the data, right? They're quite efficient at migrating pretty long term. We had shown in this paper that this is density dependent. So if the more cells you have, the more efficient they migrate outwards, which obeys to what you expect from self-generated gradients. But without showing you all the data, what was surprising for this is that these cells here, these T cells that are in the edge, they cannot do anything on their own. If you don't have the blue cells, these red cells here are not going to do anything. They're just going to stay there and not move. So there was already something quite interesting, right? Where if you look at these T cells, they're not able to do anything on their own. But if you combine them with another cell type, not only can they migrate outward super efficiently, if anything, they migrate more efficiently than the blue cells, right? Which is not completely trivial, right? I mean, it's sort of an interesting pattern already. So what do we write? So I've introduced basically exactly this equation. So it's essentially just a multi-component variation, and we've done a number of sort of theoretical variation of this, but I'll only show one, where essentially you recognize the equation from what I wrote, right? So we write sort of there's different densities, there's different populations of cells I. So for each population of cells, you write an equation for its density rho I, and you have a diffusive term and an advective term. So every cell can have different diffusivities and different chemotactic efficiencies chi, and we say that they do relative gradient sensing, and that's based, for instance, on the fact of if Michael 6 puts cells on a pre-pattern linear versus exponential gradient, cells seems to have a pre-constant velocity on an exponential gradient, which is consistent with relative gradient sensing. So if you make a gradient, if you pre-pattern a gradient, if you impose a gradient, cells are able to migrate, but not really more efficiently than basically the migration efficiency is basically the same whether you pre-make the gradient or whether you make a uniform field. But they can migrate if you engineer a gradient. Yes. Is that your question? Sorry. Any other questions? No. Yeah.
Sensing also degrades usually the sensing means binding to some receptor and then usually turnover. So you also would eat up even if so you say it's less eating than.
Yeah, so it seems like not. It seems like there's some ways to. So in this kind of GPCR signaling, there's the T cells seem to be able to sense. It could be that they eat very little and sort of negligibly little, but they don't seem to eat almost anything basically. So there seems to be different sort of efficiencies. It might not be completely binary, but they don't seem to eat up very much. No, that. The receptors, yes, but the question is whether your receptors take up the ligand with you or not. They could also get sort of internalized without the ligands. Yeah. No, this was surprising. I mean, this was surprising. We didn't completely expect it that there would be different cell populations with completely different sort of ability to sense and degrade. All right. And then you just have a diffusion equation on your chemoattractant A, right? Which essentially you can write different versions of this. Essentially here we say that the consumer degrades, so it's exactly the term that I've shown you before, and you can see that sort of the sensor doesn't feed back on the attractor. So basically, what Merman showed is that you can do quite a bit of analytics on this. You can do quite a bit of simulation. Essentially, what everything boils down to is two rescale parameter. One, which is essentially the chemotactic efficiency of the consumer, and that makes sense. That's exactly the term that I've shown you before for a single component, right? Because to get sort of chemotactic front, you need Chi-C to be larger than Chi-D than D. It's exactly the thing. So that's the ratio that we've seen before, right? That dictates whether you have traveling fronts to begin with. But then what's really important, what turns out to be really important, is the relative ability, the relative ability of both cells to do sensing. And why is that? Actually, you have a very sharp transition. So if you look, for instance, at the relative front velocity, for instance, you can ask a very simple question: can my sensor keep up with my consumers? And it turns out that there's a very sharp transition. Essentially, if your sensors are a tiny bit less efficient than your consumers, essentially they fall off infinitely fast. So essentially, what you have is essentially if you're sorry, maybe let me just show this one. It's a bit easier. This is thing 102. If your sensors are a little bit less efficient than your consumers, they're going to fall infinitely far behind. And that sort of makes sense, right? Because they cannot shape the gradient by themselves. So they have to be at least as efficient as a consumer to follow them. Otherwise, they're going to fall a little bit behind, which is a shallower gradient, which makes them fall behind, which is an even shallower gradient. So there's a huge cliff. So there's a huge cliff for a ratio of Ks over Kc minus below 1. If it's just even a tiny bit below 1, your sensors are going to fall infinitely far behind, essentially, right? So that's what's expressed here. You have tumors that form a pulse at the edge of the thing, and it's going to fall far away, and your sensors are going to be left behind. Then, if your sensors are much more efficient than your consumers, if this is much larger than 1, they're going to be in front. Sorry, let me just. Sorry, there's a problem with the animation. So if your sensors are much more efficient than your consumers, they can actually go in front, right? So they're going to form a peak ahead of the consumers, but their speed is still bounded by the consumers, right? Because as these sensors, which are very efficient, start going in front, they bump against an invisible wall, which is the fact that if you go too far away from the consumer, your gradient is flat, right? So basically, from this analysis, it already makes sense why your sensors, even though they're unable to do anything on their own, are ahead of the consumers, because it's a bit of a bimodal choice, right? Either you fall infinitely far behind, or if you're more efficient, you're going to be ahead, and how much ahead you are is a readout of how more efficient you are, essentially. You can actually get quite a bit of behind this. Beyond this, you can actually understand quite a lot of things analytically based on these expressions. For instance, you can derive some analytics for the speed of the velocity. These pulses are all exponential, and the decay function of this exponential tells you something about diffusion coefficient. So we can do quite a bit of analytics about this. And the cool thing with this is actually because the cool thing about this is actually we can sort of extract almost all of the parameters independently. So for instance, the diffusion of a chemokine, the diffusion of the chemoattractant, we got through FRAP, right? So you can bleach the fluorescent molecules and see how quickly they diffuse, and that gives you some value of your chemoattractant. You can also look at single cell diffusion as single cell motility. If you observe single cells, you can look at how much they move forward. It gives you the diffusion coefficient of your cells. And through the velocity of the wave, you can also get, and the separation of the peaks, you can also get your chemotactic coefficients. And so it turns out that actually through sort of a number of independent measurements, we could constrain pretty well all of the important ratios of our theory. And so what this predicts essentially is it predicts that the consumer forms a front that is essentially propagating at constant velocity, and that the sensors form a peak, which is a little bit ahead of the front, but with some very specific, with some really quite specific shapes. And so you can actually test that in the experiment. So you can, I mean, you can look at the experiment that I've shown you before, And you can see that, of course, the experiments are a little bit more noisy, right? But you can see that the key features of it are pretty good, pretty well reproduced, in the sense that your consumers form a traveling front that has a pretty constant shape and moves at a pretty constant velocity, while your sensors form a peak, which is ahead of the front, with, again, it's a bit hard. I mean, from this, it's pretty hard to say it's similar, but which has a pretty constant shape. What you can do a bit better is you can sort of essentially, because it's a traveling traveling wave, you can essentially go in a co-moving frame. So what you can do is you can first of all, in a model, center everything on the position of the peak. So I'm centering at every time point over the position of the peak, and I can get this way my average profile for my consumers and my sensors, which recapitulate the shape that I showed you before. And you can do the same transformation for the data. And it shows, and this actually reveals that this solitary, I mean, this kind of solitary wave propagation is pretty good, because if you look at the thick line, this is the average, and each individual line is one individual time point. So basically, the fact that there's not a huge amount, I mean, there's not that much dispersion around the average, means that if you rescale every different time point around 0, you get some pretty sort of self-similar profile for your density, right? And if you look at this, you see that the sort of the consumers, the dendritic cells are kind of a front, and the sensors are quite a back. You can actually do, and I don't want to enter too much into the details of all of this. You can do quite a number of predictions. For instance, you predict that this decays exponentially with much broader tail than this one, because this is controlled by diffusion, by the diffusion. So you predict that the exponential decay here is 3 times the exponential decay of this one, and it's also very close to what you see in the data, for instance. So there's quite a lot of features of the data that you can understand from this really, really simple Keller-Zegel type model, which actually seems to validate that you can understand quite a bit about these interactions and the presence of a traveling wave through this. What I want to ask, I mean, beyond this though, one of the things that interests me a lot these days is also to think about design principles. Like you can, sure, I mean, it's nice, you can fit the data, you can explain this with a simple model, there's some self-similar solution, very nice, and everything, but you might say, okay, why do I have these parameters, right? Is there any optimality criteria that this corresponds to, right? And in particular, if you think about it, there's a few things that are a bit surprising. Like, for instance, if what I've told you in a system, diffusion is not very good, right? I mean, if you want to have a very good sort of chemotactic front, diffusion is your enemy. So why do cells diffuse, for instance? Why do they have sort of strongly random polarity? And if you look at the ratio, for instance, of your advection to your diffusion, it's below 1, it's above 1 as it should be, but it's not, it's not a 10, it's not 100, it's not 1,000, right? It's actually relatively close. If you look at this ratio again, this has to be larger than 1, otherwise your sensor would fall behind. But again, it's not much larger than 1, right? It's sort of pretty similar. And again, these ratios, I mean, you can see that all of these ratios are close to unity, which they don't have to be, right? And we just wonder, like, why is that? So, yeah.
Experiment where you basically have like an explant, so to say, and then the cells are coming out of this.
Yeah, exactly.
So it's just a big distribution. Now, if I think about a loop node or something like this, it might be better.
Yeah.
So if there's matrix and the chemotrix becomes trapped somewhere.
Yeah, I mean, there are some. So Michael Six is the expert on this. There is something that blew my mind, which I find super cool, is that one experiment that people have done is they inject. So you can inject immune cells anywhere in, let's say, in the limb of a mouse, and you can see how efficient they are at getting to the lymph node. And that's huge, right? They have to travel sort of centimeters. And what people have found essentially is that the more cells you put on a lymph node, the more in proportion you get. The more cells you put in the limb, for instance, the more you get in the lymph node. You see what I mean? So if you put a certain concentration of cells, let's say if you put 1,000 cells, you're going to get 10% that are going to get to the lymph node. But if you put 1,000,000 cells, there's going to be 50% that gets on the lymph node. And that shows that's something that shows in vivo that there is some collective features of this, that essentially sort of putting many cells is more efficient than putting few cells, which I think is a super cool validation that there is some collective effects in this kind of, even in vivo actually. which again, maybe not in the lymph node, but still to migrate to the lymph node, you need to migrate pretty far, you know, in lymphatic networks and things like that. Yes. So that's the hardest one to measure. This is based actually on. I mean, you can essentially do it. You can do it two ways. So one is you can do the experiments without any cells. So you know, for instance, if you get it only with the consumers, you can measure it independently without the sensors. The chi shows up in the speed of the migration, and the relative chi's also show up in how far are the peaks of the sensors. You know, how far are the sensors ahead of the consumers, and so you can measure it through different ways, and they agree with each other. That's basically the point. Yeah. All right. So the question is, the question that we had is, why are all these parameters, all of these parameters sort of close to unity? And the idea that we had essentially is that if you think about these immune cells, they have to migrate. So what they have to do is they have to migrate quite efficiently, right, towards the lymph node or towards an inflammation. But what is known also is that they like to interact physically, because they also sort of exchange informations, antigens, and everything. So they also want to interact physically. And so if you imagine this, for instance, if you make your sensors if you make your sensors like 10 times more efficient than your consumers, your sensors are going to form a peak that's way above, that's super far ahead of the sensors, right? You can imagine something like this. And so if your sensors are super efficient, they're going to be completely spatially non-colocalized with the consumers, right? So if your sensors are too efficient, you're going to be very good at migrating, but you're going to be very bad at colocalization. And similarly, if you think about diffusion is bad for migration, but diffusion is good for colocalization, right? Because if you put diffusion to 0, that's going to be very good to make sort of, you know, delta functions sort of moving forwards, but they're going to be completely spatially uncorrelated. So the idea that we have essentially is that Mehmet did rather is essentially to derive some kind of phase diagram where we look at these two things, and essentially to try to optimize both the fact that you should migrate in an efficient way, but you should also be colocalized. And what it shows up again is that if you think about it theoretically, you want all of your parameters to be relatively close to unity, right? Because essentially all of your parameters that make your migration very efficient make you poorly colocalized. And so the optimum of colocalization is this thing in blue here that you want to be. So here, for instance, if you're very efficient at migrating, if your sensors are very efficient at migrating, they're going to be very good, but they're going to be very poorly colocalized. And as I said, here, for instance, if your sensors are not so efficient, so because they're going to fall very far behind. There's like quite a thin valley of an optimum where you're robustly coupled, but you're also colocalized. And if you put sort of the experimental data sets, so this is the inferred region of parameter space that we found, and it turns out that it sits pretty close to this sort of optimum as a trade-off. So basically, what we're sort of wrapping up in this paper and proposing is essentially that maybe this is some design principle of coupling of populations where you want to optimize where all of your parameters are intermediate, because you want to optimize both for robust migration and sort of colocalized co-migration, basically. And yeah, I'm not going to enter into anything else actually, because I don't have the time, so I can just start. I can get any questions if there's anything left. Yeah, that's so. I don't know much about immune. I mean, about immune response. So I guess there are different cell population that comes from hematopoietic stems. I mean, I would think that there's like some hematopoietic stem cells, and there's like a bunch of differentiation. But I don't know how far they are in the genealogy of hematopoiesis. I have to say, I don't know much about the genesis of cell populations. No, I think they're pretty differentiated. So they're, I think at this point they're essentially sort of no, they're independent populations. Yes, I think so at least. Yeah. Sorry, there was another question. Yeah. So, in that paper, they don't pass it to the continuum limit. So what they do in this paper is that they simulate agents, and then they sort of so essentially they simulate sort of the diffusion equation with sinc terms that are your particles that are moving, and they take the diffusion they take diffusion to be quasi-static, so they take diffusion to be equilibrated. And then you have your particles evolving in your quasi-static diffusible field. And so that's what they do. They don't pass it to the continuum limit, actually. Yeah, there were some. Yeah. No, so you're right. We didn't put any. This would be the next thing. We didn't put any interaction terms. So, for instance, we could put some cross diffusion. We could say that there are some physical interactions between different cell types. We didn't put it in there. The reason, there's multiple reasons we didn't put it in there. For starters, because actually there's some experiments where you can make the red cells being neither sensor or consumers. So you can make them essentially completely passive. They cannot eat, they cannot sense. And in that case, they stay completely, they don't move at all. So they're not, it's not that they're sort of, advected by the others. There's no, there's probably some interactions, but what's for sure is that the red cells, the sensors also need to actively migrate. They're not just sort of, dragged like this. So it's not, at least it's not physical interactions being dominant. So that's why what we think is that it's really an interaction through a diffusible field where the consumers sort of eat the gradients, and so they make a gradient of diffusible molecules that the sensor cell can sense. So we really think it's a mode. It's a way where your populations of cells are coupled not through physical contacts, but just because they manipulate the chemoattractant field. But we should more systematically look at physical interaction between cells. This would be we would have to add some cross advection term or something like this, and we probably should do it again. I think there's some experimental evidence that it's not dominant, but we should do it for completeness because it's interesting. Yeah, sorry. No, so we do everything. So the chemoattractant is uniform, and the cells are on one side. So you this. Yeah, exactly. Well, there's a delta. There's a heavy side. The cells are an aggregate, so there's an heavy side on one side. Yeah, and then yeah. Oh yeah. Yeah, so I don't think there would be pattern formation because we're in a limit where the cell eats their chemoattractant, and not. I think pattern formation requires for them to produce their own chemoattractant also to get advected. So Myco6 is trying to make these cells overexpress their attractant to get into some dictyostelium limit where they aggregate. So which would be cool. For now, the chemoattractants is something that we provide and that the cells eat, so they're not made by the cells. I think to get in the pattern formation regime, you would have to invert that sign, basically. Yeah. Sorry.
I was wondering whether you think this is an example of leader follower, or what do you mean?
Yeah, it's probably a type of leader follower where but I think the point is that you don't need any active sort of leader follower interactions. If it's not really leader follower because it's the cells in the back that guide the cells in the front, right? So it's these ones are only in the front because they're faster, because they must be faster, otherwise they would fall behind. But if anything, these are pushed. It's more a pushing mechanism, right? Where these, it's not physically pushing, but it's these ones that make a gradient, that these ones move forward. So if anything, it's funny. It could look that way. It could look that way from the position, but the relative position of the cell is misleading. It's actually the reverse causality, right? Which is funny. Yeah.
One cell becomes the. So I think the terminology wants to show also this. Someone is elected, right? But here they are already different functions.
No. Leo, you had some question? I don't know. I mean, you don't have, I mean, you have your C term, right? So you can't integrate it once. I mean, you have to, yeah. I don't think there's an, I think you can get some analytical solutions from very specific velocities. So I think there's some exact solutions if your velocity is 5 over square root of 6. There's some really, I mean, there's some really weird sort of cases where you can sort of find analytical solutions. Otherwise, I don't think, but in some specific case, right? I think in some specific velocities, I think that's what I remember. But yeah. I mean, people have done. I mean, people. I mean, for instance, like the fact that it's I mean, a lot of people in the physics literature, because it's mapped on sort of branching random walks, right? So for instance, people have been mostly interested more looking at corrections because there's some logarithmic corrections to your sort of constant velocities. So I think people have been a bit. I've also looked at this quite a bit because of that, yeah. I mean, that's a classical result. Yeah, that's this all.
You can set the velocity and then it will, like, what happens to a velocity is bigger than the critical one?
So this minimal one, I mean, if you start from a delta function, for instance, the minimal one is the one that gets selected. I'm not an expert on this. The thing is that I think the contrary to Köhler-Zegel is a lot more robust. The problem with the sort of Fisher is that because there's some exponential growth term, it's very sensitive to your initial condition. For instance, if your initial condition is not a delta function, but it's something with fat tails, then everything grows exponentially and you don't have even any, so which is something that doesn't happen. It's a problem of growth that doesn't happen. And so in that sense, your Köhler-Zegel equations are much more robust to initial conditions than Fisher. But I'm not an expert on this, but that's the general solution. Yeah.
If the cell has a larger chi, then it means that it can sense smaller gradients?
Yeah, exactly. It's an efficiency. Yeah, it's an efficiency of it's how you translate a given gradient to a given velocity. So it's like an efficiency of chemotaxis. Exactly. Yeah. It's the simplest term to. I mean, you know, I mean, there's a big assumption here that we take it constant, right? In principle, you could say that, for instance, your receptors are saturated, so you could make your chi sort of. I mean, you could make your chi A dependent, for instance. So we didn't. I mean, we didn't consider this because it's just complexifies a bit everything. But you could make it. That's the principal thing you would do. You wouldn't get it out of the parentheses. You would just write some chi I of A that would vary. I mean, people in single cell chemotaxis have thought. I mean, there is. I mean, this I go to the consumer level. People in single cell chemotaxis have thought a lot about, you know, like about, you know, which kind of mechanisms give rise to which kind of gradient sensing. I mean, this is something I'm not specialist about, but there is a lot of literature on this. Yeah. All right. All right. Cool. Thank you very much.